Hello students, this is Ashta K. Sanush, Assistant Professor, Department of Microbiology, EMA College of Arts and Science. Now, in this lecture, we'll talk about how exactly this VDJ recombination occurs. We'll talk about hypersomatic mutation and antibody class switching. Okay, so talking about the VDJ recombination, it actually has 7 steps. Let me just write it down. Maybe I'll write it down here. That's better. One is, first step is it recognition of signal sequence by recombinase. Second step, cleavage of one strand by rag rag1 and 2. Third step involves the esterification reaction by the rag1 and 2. The fourth step is trimming. Okay, it's not trimming, actually it is a nicking of hairpin loop by endonuclease enzyme, endonuclease. Fifth step, here the trimming actually occurs, trimming of nucleotide by exonucleases. Then you have the nucleotide addition. Then you have repair by DSBR enzyme. So these are the steps which are involved in the recombination of different regions. Now let's see what actually happens here. As we know that there are different regions present, right? That is you have a V region, you have a D region, and you have G region. V stands for variable region, D for diversity, and J for joining. So initially, I told you like we have genes coming from mother and from father. Only one gene is expressed. So in this case, let's say it's mother's, then the gene should have genes for immunoglobulin, right? So initially it should have genes for heavy chain. If it has genes for heavy chain, it will make heavy chain. And if it does not have, it will undergo apoptosis. Okay. Then once the heavy chains are made, it will have to make light chain. Light chain kappa will be given first preference. Then if kappa, it does not have genes for kappa, only then it will go for lambda, right? And if it has gene for kappa, then it will make kappa light chain. And if it does not have, it will make, see whether it has genes for lambda, then it will make lambda light chain. And if it does not have both, in that case, it will undergo apoptosis. Okay. Now, and we also know heavy chain, it is V, D, and J. Light chain, it is V and J. And this both followed by constant, right? Okay. Now, in this VDJ recombination, you have gene pool of variable region. Then you have for diversity. Then you have for joining, right? So in my previous video, I told you that these both are going to initially, the combination of these two occurs, one part of this and this is going to get joined, and then the variable region also, one part of variable region comes and join. So in order to occur the joining to take place, it needs something called RSS, okay, which is known as recognition signal sequence. So that is the recognition of signal sequences. Okay. So this RSS, if you actually zoom in, it has got a structure where it has an heptamer. Heptamer means how many nucleotides? It is 7 nucleotide. Okay. By 12 or 22 base pair, followed by again 9 nanomer, that is 9 nucleotide. Okay, so in few books you can see instead of 22 it might be 23. Okay, so the way this V and D or D and J is going to come, then it is based on a rule called 12-22 rule. So what actually happens when you actually, let me just say, take V1, V2, V3, and so on, Vn. So you don't want these members of the same region to join with each other. So in order to avoid that, a rule called 12-22 base pair rule is followed. That is, if this variable region has 12 nucleotides, and then the diversity side should have 22 at one side, okay, and if it needs to join with G, it should have 12 here, and this side should be 22. That means, okay, let me tell you, this variable region, this RSS comes and binds to its one side, as the D has to join both with V and J, it will have two RSS, okay, and the J will have RSS to this side. So if this has 12 base pair, this is side of the diversity region, D region should have 22 base, and if this has 22, obviously this will be 12, and here it will be 22. Only then they can actually combine. Okay, and another thing which I need to tell you is apart from RSS, there is something called RAG1 and RAG2, which are nothing but recombinase activation gene. These RAG genes will be always found in associated with this RSS. These are RAG genes. They will be found in associated with RSS, this both sides. Okay, this is how actually it will be present. Now, following the rule, one part of V and one part of D, then one part of D with another part of J is going to combine, right? So let's consider, as I told you in initial lecture, these two will be combining first, that is the D and J. So let's consider that D2, so D2 and J3 is going to combine. Okay, so let's just consider initial D and J. So you have D2, then you have RSS, and then you have the RAG, and here you have RSS, RAG, and then you have J1 or J2, and this is your DNA. It is all in DNA, right? This is the DNA. Now, once you actually bring these two parts together, okay, the intertwining the DNA, it will be forming a loop. Okay, it will form a loop. Just consider you have a thread and you just start pushing it from both sides. Now what will happen? It is going to form a loop. Okay, similarly, this will form a loop, and what will happen? The DNA between them forms a loop, and this gets cut off. Okay. Either it can just make a single loop. Sometimes there is another combination, which is a double loop. Apart from single, it can also make double loop. So these part will be actually cut off. Now, let's just consider this is a double-stranded DNA and whole things are happening in between. And once this loop has been cut out, you just have only a small thing which will be present here. Okay, a small region alone will be present. A part of RAG gene will be present. Okay, just consider this part alone will be present. Now, what will happen? A RAG gene will make a nick. Okay, it just makes a nick. Now, once it makes a nick here in this particular first DNA strand, it will release OH ions. Okay, now these OH ions will make a nick in the second strand. Get it? The RAG gene, after this loop has been cut off, a part of RAG gene will be present. So this RAG gene makes a nick in one of the DNA strand because it cuts one of them, then it releases hydrogen ions, OH group. Okay, that makes a cut in the second strand of the DNA. Now, once it makes a cut, what will happen? It breaks, actually esterification occurs. Esterification occurs. And the opposite strand, that is this strand, and this is going to cut the DNA. Okay, this is what happens. And similarly, even here in the J region, there's a J and there's a D region, right? Similarly, the RAG cuts and it's going to make a nick here, okay? So what will happen? So I hope you get it clear that once you try to bring this two region closer, in between the intertwining DNA will be forming a loop and it just cuts off. A small portion of RAG will be present. Then so what it actually does, it is going to make a nick in one of the strands of the DNA which releases LOH, that is hydroxyl hydrogen group. Now these hydrogen groups go and make a nick in the other strand, which causes the esterification, and it actually nicks the opposite strand. Similarly, the same process happens in the J region. So now you have, so this is where we are reached. If you see the recognition, okay, this is the part recognition. Then you have the cleavage of one strand by rag 1 and 2, trans-esterification reaction by rag 1 and 2. Okay, now nicking of the hairpin loop. Let's see what happens then. Here, okay. So once the nick is formed, what will happen? You will have your rag, and just consider this part, and you will have a small part of rag present here. Similarly, in the D region and the J region also, a small part of rag also will be present here. Rag with the loop remained after the cutting of the double-stranded DNA. Now, in between, you have, once you have the rest of the DNA, okay, the DNA will be present, right? The rest of the DNA which is present here, okay, this is known as the intervening DNA. So this is how it happens, and what this gets actually removed. And let me just now focus only to that particular D2 or and small part of rag, and you have J1, whatever you have a part here. Now this D2 and J1 are very close, and a small hair loop is still present there, right? To connect them, okay, how you need to connect these two. There is something called Artemis. Okay, this is an endonuclease. It comes and nick or open up the loop. Opening, what it does, it is going to just open this loop. Okay, it is going to open the loop. Open slightly uneven. It doesn't mean that it is going to just be the perfectly, it is going to break it. No, it is not going to be perfectly. This is going to just randomly, it is going to cut here. Okay, now, so what will happen? So randomly, when it is going to cut here, there is another enzyme which is called T. So this T, as I told you, the cut is made in such a way that it will not be a perfect cut. Now there is another enzyme, as I mentioned, that is a TDT, which is called as a terminal deoxydeaminase transferase or polymerase. So what actually this thing, it does it, if you just consider this as D and this as J, okay, so it will not be even sometimes, okay, so just like this. It is going to add nucleotide to this, okay, it's going to add nucleotide to this region, okay. So there will be a junctional diversity created, because it's not like, just suppose while randomly, if you see that D2 and J1 is making a nick here, if you take another recombination which is occurring between D and J, maybe D1 and J3, it will actually make a nick in some other point, and nucleotide gets added to that particular place. So there will be a variation on where this amino acids or nucleotide will be added. Okay, there will be no specificity present here. So this is the reason why there is a diversity created. Now this TDT or the polymerase, it can inject or it can remove some of the nucleotide. Sometimes it can also remove if it is not a proper base pairing occurring. It can also remove certain nucleotide from it. Okay, so it what does ultimately what it does is it is going to change the DNA, right? It is going to change the DNA, right? Now what will happen, another enzyme after TDT it is going to come is the ligase. Now the nucleotide has been added, okay, which also is known as P and N nucleotide, and there will be a diversity created that is called as the junction diversity. Now what will happen, another enzyme called ligase is going to come. It is going to close the nick, the double-stranded DNA repair enzyme. The last part is this double-stranded DNA repair enzyme is going to come and repair if there is any mismatching of nucleotides present. Okay, now I hope you understood this. Okay, let me just wind it up and then let me start the constant region C part in separate video. Okay, just consider you need to bring the V, D, and J has to be combined here. So what happens, it needs RSS and RAG, okay, and it follows a rule which is called as 12-22 rule. So what happens in this is that the RSS, that is the recognition signal sequences, along with the RAG, okay, so you need to bring these two. First, initially, you need to recombination of this D and G takes place. So when it actually brings the part which is present in between, part which is present in between these two will be cut off in such a way that it is a small part of rag is going to be present. Right now, this rag, what it does, as you can consider all these things happening in double-stranded DNA, rag is going to make a first, it is going to make a nick in one of the strand, releasing the OH ions. Okay, this go and cut the next strand of the DNA, okay, which is called esterification. Now, once it cuts, okay, this small part of rag will be present. So this will be your D and this will be your J. So small part of rag still are present. Now, what will happen once the other intertwining DNA will be cut off from it, okay, only this two part will be present. And there is another enzyme which is an endonuclease, which is called Artemis. It is going to cut open this loop and it in such a way that it will not be equal, sometimes till here and the rest will be from here. Second strand, maybe it's like this and this will not be, it will not be anything specific, it will be random, right? So what will happen, there's another enzyme which is called as TDT. It is going to add nucleotides here. Once it adds nucleotide, the ligase is going to come and it is going to join it. And if there is any kind of mismatching occurring here, the repair enzyme, double-stranded repair enzyme is going to come and it is going to repair that particular, either it is going to remove it and it is going to uh, all the process is going to bring up both this region together. And similarly, the same process happens once these two gets joined, it is going to go and join with the V region and the whole same thing is going to process. Uh, repeat itself. So as I mentioned, the seventh stage initially is the recognition of signal sequences. Then the RAG1 and RAG2 is going to cleave. There occurs a transesterification reaction, the nicking of the hairpin loop by endonucleases Artemis. and then trimming of the nucleotide by exonucleases, that is TDT, that will also add the nucleotide, creating a PN nucleotide or junction diversity, and thereby if there is still any mismatching occurring, repair enzymes is going to come and repair that particular nucleotide. Okay, now the ligase, and this is the whole process by which this VDJ region can combine and act, and this is one of the factor which cause the diversity in the antibody. Okay, thank you.